Hi guys, let's get started. And in the last week, we learned the basics of genetics and we saw different types of vaccines and CRISPR technology, which helped us to detect COVID. And then we concluded the last module with bioinformatics. Now let's shift our track to lab-grown organs. I am Samiksha MS, and I'm a bioengineering student at IIT Madras. I'm fascinated by the potential of bioengineering to improve human lives. And in this module, I'm excited to share my knowledge about this exciting area of research with you all. Let's embark on this journey and discover the possibilities of becoming immortality's apprentice through lab grown organs. So, let me start my presentation. Is is the slides visible? You can respond in the chat. Yes. So these are the topics which we'll be looking today. First, we'll be discussing First, we'll be discussing about the transplant statistics in India and what do the hospitals do. And then we'll be discussing the importance of genetics in organ donation. And we'll also discuss about the blood types. Then we'll be having a brief introduction about lab grown organs. And we'll dive deep into stem cells and its types and its potential for organ creation. Finally, we'll conclude this session by discussing the protocols for creating iPSCs. So, are you guys ready? If yes, put +1 in the chat or give me a thumbs up. Yes, this is. So, let's get started. Let's discuss about kidney failure and its transplant statistics because it's the most common in India. As you can see here, there is a very huge gap between the people needing kidney transplants and the donors available for it. Estimates suggest that around 1.8 lakh people suffer from kidney failure every year, and this translates to roughly about 15,000 people per month. But the number of transplants performed are only 6,000, and this creates a very big gap between the demand and supply. Now let's discuss what do the hospitals do exactly when it comes to organ transplantation. The first process is pre-surgery evaluation where the hospitals thoroughly ensure the both recipients and the donors to ensure compatibility and suitability for the surgery. So, do you guys know what test they take? Any idea? You can answer in the chat. What test do they take primarily? Microtoxicity tests. Yes, compatible biotype, blood types. Yes. Yes, guys, you're right. This involves blood tests, tissue typing. Yes, Gaurija, you're right. And they take imaging scans as well. So we'll be discussing about blood types and tissue typing later in this session in much more detail. So let's move on to the next step, which is pre-surgery preparation. Here, the recipient undergoes dialysis if their kidneys are no longer functioning well. Again, now the question is, what is dialysis? Do you guys have any idea about it? Yes. Oh, nitrogen is waste. Yes, it's right. It's basically a substitute for kidney if it's not functioning properly, but it's not feasible for a long run. It's a process which purifies the blood, which is the job of the kidney. Now let's move on to the second steps which the hospitals perform, which is surgery. And in surgery, there are two types. The first type of surgery is the donor surgery, uh, in which if it's a living donor transplant, surgeons remove the healthy kidney through an open incision. And the second type of surgery is the recipient surgery, where the surgeon creates an incision in the lower abdomen and they carefully position the donor's kidney into it. Then they connect the blood vessels of the new kidney to the recipient's blood vessels and they connect the ureter as well. So, what happens after surgery? The recipient stays in the hospital for monitoring and to ensure that the new kidneys functions well. The hospital also provides anti-rejection medications to prevent the body from attacking the new kidney. So we'll be discussing about the rejection mechanism in in a few time. And then the hospital schedules regular follow-up appointments as well. So this is what basically hospitals do when it comes to an organ transplant. Now, why are shared genetics important? Why are family members always mostly preferred as donors for organ donation? So here is where we discuss about tissue typing. When we receive an organ transplant, our body recognizes it as foreign tissue, and our immune system is designed to fight off the invaders so it can attack the new organ, which might lead to rejection. So, this is where the shared genetics come in. There's something called as human leukocyte antigen, which is a system of genes that determine how our immune system identifies self from non self. The more closely our HLA markers match to the donors, the less likely our body is to reject the organ. So, by matching the HLA markers closely, doctors can significantly reduce the risk of rejection. In essence, shared genetics helps us find organs that are more compatible with the recipient's immune system, which can lead to a higher rate for transplants. Now, how does genetics influence blood type? What makes blood type unique? Our blood type is entirely determined by genetics, specifically by a set of genes that control the presence or absence of certain molecules on the surface of our RBCs. Do you guys know the name of those molecules? What are those molecules which are present on our RBCs that determine the blood type? Antigens. Yes, yes. Yes, you're right, guys. So antigens are the tiny proteins which are present on the surface of our RBCs. Now there is a gene called as ABO gene, and that gene determines which blood type we'll be having. And this gene have three alleles, which are A, B, and O alleles. Do you guys have any idea about what alleles are? Do you guys understand the meaning of alleles? No. So let me explain it to you now. Can you guys see the whiteboard? Okay. So let's discuss the concept of alleles with the help of a simple trait which is eye color. So let's draw chromosomes first. Let's say you got this chromosome from your dad and this from your mom. So here is a gene which codes for eye colors. Chromosomes are nothing but condensed form of DNA, right? And DNA contains genes. So let's say this is the part where the genes for eye color is present. Now, your father has the gene which codes for brown eyes. Let's denote it by capital B. And your mom carries the genes which codes for blue eyes. Let's denote it by small b. So when they come together, they compete with each other, and finally the dominant one gets expressed. So as as we see here, the trait is the eye color, but there are two variations in it. One is the brown eye color and the other one is blue eye color. Guys, I you can see my presentation, right? Echo, you can try refreshing the page once. So this gene this gene's trait is eye color, but it comes in two variations, which is brown eyes and the other one is blue eyes. So this variation for a particular trait or for a particular gene is called as alleles. Do you guys get it? Do you guys get the meaning of alleles? Okay, wait. Guys, I'll explain it without whiteboard itself. See, there is a gene which codes for eye color. Let's say your father's gene codes for brown eyes and your mom's gene code for blue eyes. When they come together, they compete with each other, but finally the dominant one gets expressed, right? What we saw here is a particular trait which is eye color, but that comes in two variations, which is brown eyes and the next one is blue eyes. So these variations in a particular trait or a particular gene is known as alleles. Do you guys get it? Okay. So let's continue our discussion uh, with blood types. The analogy is similar for blood types as well. So wait, let me present first. Is my screen visible now? Okay. Okay, let's continue our discussion then. You can relate the concept of eye color to the blood type as well. Here the gene is the ABO gene, and here there are three types of alleles, which is A, B, and O. A allele creates A antigen and B allele creates B antigen. But O allele does not create any antigens on its surface. So depending, how does this help in determining the blood type combinations? So, depending on the combination of allele we inherit from our parents, will have a specific blood type. For example, if you inherit A allele from your father and B allele from your mother, you will have AB blood type. Why AB? Why not only A or only B? Because A and B are codominant, which we have discussed earlier. But if you have O blood type, then you must have inherited O allele from both your parents because this O allele is a recessive one. So we can conclude that the specific alleles which we inherit from our parents dictate the type of antigens which are present on our RBCs, and then that ultimately decides our blood type. Now, many of us have heard something called as B positive and B negative, this positive and negative types. What do you think determines this variations? Anyone in the chat? RH factor. Yes, guys, you're right. You're right. No, see, I think you didn't understand the concept of blood typing well. Both your dad's and mom's gene determine your blood type. It depends on whether their alleles are dominant or recessive. If your father, I'll explain this with another example. Let's say your dad has A allele, and we know that A allele is dominant, and your mom has O allele, which is recessive. But when they both come together, finally the A allele wins the fight, right? Because it's dominant, and only that trait is expressed in you. The O allele becomes recessive in you. That does not get expressed. Did you get it, Sugi? Did you get it? Okay. So there is one more factor. Yes, you guys are right. That is called as RH factor or the rhesus factor. So this determine the positive or negative type of blood. This is a separate protein found on the RBCs, and people who have these proteins are Rh positive, while people who doesn't have it are known as Rh negative. For example, B positive and B negative. The Rh factor is also inherited through genes, but it follows a much simpler dominant and recessive pattern, which is Rh positive is dominant and negative is recessive. So by so I think you have understood this concept. Do you have any doubts in this, guys? Any doubts? Yes, Rh factor is also a type of protein. It is a protein that is present on the surface of RBCs. What do you mean by a separate protein? Arushi? Yes, yes, it is separate from antigens only. Is it clear now? Okay. So I think we can move on to our next concept. So by understanding these genetic factors, we can not only predict the blood type but also we can determine the blood compatibility for transfusions and organ transplants. But how? Checking the blood type compatibility is the first and most crucial step when it comes to organ transplantations. This must be done to prevent rapid and severe immune reaction which happens in our body. So there are four blood types. O blood type is called as the universal donor because they can donate to anyone. And AB is called the universal recipient. And there are other specific rules which can dictate what blood types we can donate to others. So we'll be discussing about more about it in few time. But before that, let us discuss what happens if the wrong type of blood is infused. We already discussed that immune reaction will be triggered, but do you guys have any idea of what exactly happens? What exactly happens within the cells? Is there a name for it? Do you guys know the name? Yes, agglutination. You're right. Kushi. You're right, Kushi. So let us discuss in detail about it. So there are generally many antibodies and antigens present in our body, but when we come to the context of RBCs, there are two antigens which we yes, Gaurija. When we come to the context of RBCs, there are two antigens, which is A and B, which we have discussed earlier. But to understand the concept of rejection, we need to know another factor as well, which is known as antibodies. So these are proteins produced by our immune system to fight off any invaders such as bacteria, virus, or any foreign particles. So when we see A blood type has antibodies for B blood type, which is known as anti-A antibodies, anti-B antibodies. Similarly, B blood type has the antibodies for A. So with this, can you guess what must be the type of antibodies present in AB blood type? Anti-O, anti-AB, no antibody. Yes. So there is no antibodies present for AB blood type. Yes, you guys are right. And can you guess for O blood type as well? Both. Yes, guys, you're right. Both A and B type of antibodies are present in O blood type. So we have discussed this. And yes. Uh, Let me explain the concept of agglutination with an example. I am B positive. Okay. Let's say an A positive uh, person donates me blood. So what happens? Since I'm B positive, what is the type of antibodies present within me? Anti-A antibodies. Yes. In my body, anti-A antibodies blood type is present. But when someone with A positive donates me his blood or her blood, what happens? The antigens, there will be a reaction between this anti-A antibody and the A antigen present on that RBC, and a reaction happens between them. That is, they bind together. And this process is known as agglutination, which will eventually cause the blood to form like clumps, or it is similar to blood clot. and this is a very dangerous condition. And this is why the first test which is done is blood test. You can see here there are different types of antigens present on the surface of the blood. In AB, you can see both the type of antigens, and A blood type has anti-B. You can see the color matching. You can see the color matching here. Antigen B is suitable for anti-B antibodies, so that's why they are not present in the same blood group. Did you guys get this concept? Yes. And this is the reason why O blood type can be universal donors, because they do not have any antigens on their surface. Even they can donate, see for example, they can donate to B blood type because B blood type has anti-A antibodies. But what will it go and attack? There is no antigen on the surface of RBCs, on the surface of RBCs of O blood group, right? So O blood type is compatible with all the blood types. But AB is universal recipient because there is no antibodies present in them. So even though they have antigens present on their surface, there will be nothing to come and attack those antigens. So they can receive blood from any of the blood type. Did you get this concept of compatibility, blood compatibility? Yes, you are right, UV Bari. O can give to any blood type. Okay. So what are the other factors uh, necessary for organs to be compatible? The first one is body size. Obviously, the donor's kidney and the recipient's kidney should be similar in size for the kidney to function well. Not only kidney, I'm just taking kidney for an example because at the start we discussed about kidney only, right? This is applicable for any of the organs. And the medical history of the donor is crucial as well. So, till now, do you guys have any doubts? So, if no doubts, give me a thumbs up. Guys, I think we can move to the most awaited topic, which is the concept of lab grown organs. And before that, let us see here. This is what the blood with agglutination looks like. It forms like a clump here. You can see in this pic. So let's move to lab grown organs. Imagine a future where instead of relying on organ donors, doctors can create new ones specifically for each patient. And what is the advantages of it and how can it be done? Scientists can use stem cells which have the potential to develop into any cell type. And that stem cells can be taken from a patient's own body or they can be taken from a universal donor source. So we'll discuss about stem cells in the next section of today's session. So let's wait for it. And using a patient's own cells could significantly reduce the risk of organ rejection, which is a major complication in organ transplantation nowadays. But we are continuously saying, right, rejection risk, organ rejection. But what exactly happens? We have already saw a brief introduction about it. What exactly happens? Do you guys have any idea? Universal donor source means it's similar to blood banks. Stem cell banks are also present, right? So anyone can donate their stem cells which can be used for treatments for other patients. Did you get it, Sampurna? Yes, we can do that, but in case we need someone else's stem cells, we can approach stem cell bank. And Jyotin and Sagar, you're right. Let us discuss it in a much more detail. Once the recipient's immune system recognizes the transplanted organ as foreign, it starts an attack. Because due to the differences in proteins called as human leukocyte antigen, which we have discussed earlier, the immune cells, primarily the T lymphocytes, identify the HLA mismatch and they launch an attack. They release chemicals and they target the organ cells for destruction. Now, what happens? Tissue damage. The immune system attack damages the transplanted organ's tissue, which hinders its function. And as the damage progresses, the organ's ability to perform its essential function, for example, kidney filtering its blood, is reduced, and it will lead to organ failure and it might lead to death as well if it's not identified in an early stage. Lab grown organs are in early stage of development now, but it has an enormous benefits. It could mean that patients in need of transplant, no patients in need of transplant no longer wait for their turn to get transplant, because nowadays there is a very big wait list and patients often wait for several years to get transplant, and meanwhile their health also becomes worse and worse, and this will even complicate the process of transplantation. But the help of lab grown organs, we can reduce the wait time and it can bring an end to the wait list as well. So let us see an interesting case study here. Scientists are working on a very new way to grow human organs for transplants. So what they are doing is they are transplanting stem cells from a person into an animal embryo at an early stage of its development. So this has been seen successful in mice and rats, but for humans they need a large animal. The next step is to transplant these cells into a sheep or pig embryo. And as these animals grow fast and have organs similar to the size of a human. The challenge is getting the human stem cells to become a part of the animal's organs and not to be rejected. So there are some ethical concerns in this as well because they don't want the animal to develop human characteristics. And so far, this research shows that the animal's own cell will control the development and the human cell will be a part of that organ. So this research is still in its early stage, but it has the potential to solve the organ shortage for transplants. So can you see the link here? This is an article about this case study. You can explore more about this after the session. I'll be uploading the PPTs and you can access this link from the PPTs. Yes, guys, can we move on or do you have any doubts? Uh, Dritijana, are you asking? I'm not getting your question. Can you explain it in a more better way? See, the human stem cells are not taking the control of development, right? Till now, the research is only the animal cells will control the development of the organ. And as we'll be seeing in the next section, stem cells can become into any cell type. So the human cells will also get integrated into the part of the animal, and it will also become the part of the organ. We'll get back to that later, Ritijana, or you can see more about it in the given link. So, can we move to the concept of stem cells? Yeah, it's very similar to that. Anushka, can we move on, guys? Give me a thumbs up or plus one in the chat. So, what are stem cells and what makes them special? So, there are two properties which makes these cells special, which is the self renewal and the differentiation. First, we'll discuss about the self renewal property now. So, imagine a magic well that never runs dry. It's similar to that. Unlike most normal cells in our body that have a limited number of divisions before they die, stem cells can copy themselves for an extended period. And this continuous self replication allows them to act as a sort of an internal repair system and constantly replenishing the body's supply for specialized cells. Now, the second property is differentiation. When we see the normal cells are specialists, each with a specific job. For example, the muscle cells helps us to move while the RBCs carries oxygen, but stem cells are unspecialized. They have the potential to differentiate into many different types of cells depending on the signals they receive from their environment. And this versatility makes them incredibly valuable in regenerative medicine. But before discussing the type of stem cells, let's discuss an interesting question now. So, our body is entirely made of cells and tissues, and all the cells have the same type of DNA, right? Yes or no, guys? Yes. Others? What about others? Hmm. All the cells have the same DNA, but what makes them unique? What creates different types of stem cells? No, sorry. What creates different types of cells? What is the process behind it? Do you guys have any idea? Uh, I'm not sure about this totipotency factor, but it's a much simpler factor which is known as transcription. I think Are you guys aware of transcription? Yes. This is a very simple process by which what decides what function should the uh, cell perform. See, all the cells in our body have the same DNA blueprint, but they come in many different shapes and sizes with specialized functions. The key to this difference lies in a process called as transcription, and which are controlled by a special proteins called as transcription factors. So let's discuss about it. So for those of you who doesn't know about transcription, I'll be explaining it right now. So imagine DNA as a giant recipe book. It contains all the instructions our body needs to build and function. But these instructions are too long to be used at once. These transcription factors, which we discussed now, act like chefs. They scan the entire DNA recipe book looking for specific start and stop signals that mark the beginning and the end of a specific gene. Once the gene is identified, the transcription factors call in an enzyme called as RNA polymerase. And this enzyme acts like an assistant chef which copies the gene into a much shorter and a more manageable form called as messenger RNA, called as mRNA. Then the mRNA travels out of the nucleus carrying the specific recipe to the ribosomes in the cytoplasm. And these ribosomes use the mRNA recipe to build the final product, which is called as proteins. Now, what is this process called, guys? The process where the mRNA is changed into proteins. What is this process called? Yes, guys, you're right. It's called as translation. So the but here's a twist, guys. There are different transcription factors that have different preferences for which genes they activate in the DNA. Like there are different chefs, right? You can imagine that analogy. Think of it like chefs having their favorite dishes. A muscle cell's transcription fact a muscle cell's transcription factor might favor the genes for proteins which are required or involved in contraction, while nerve cells transcription factor might activate the genes for proteins involved in sending the signals. So by selectively turning on the specific genes in different cell types, transcription factors essentially determine which proteins are made in the end, and in turn, that decides what function that cells perform. So, even though all our cells have the same DNA recipe book, the specific transcription factors choose the genes which gets transcribed and in the nucleus, which gets transcribed in the nucleus, which results in a diverse range of cellular functions. So, do you guys get it? Do you guys have any doubts till now? Give me a thumbs up if we can move on to the types of stem cells now. Okay, let's now move on to the types of stem cells. There are three types. First is embryonic stem cell, the next is adult stem cell, and the third is induced pluripotent stem cells. We'll discuss each one of them now. First, discuss about embryonic stem cells. These are derived from a very early stage of embryos, and these are the most versatile of these stem cells. They are pluripotent, which means they have the potential to differentiate into any of the body's cell types. This makes them an incredible value for the research on regenerative medicine. But their uses raise ethical considerations, and we'll not be discussing them in today's class. We'll be discussing them in the third session of this module, which will be happening on Friday. So don't miss out on any of the sessions. Now let's move on to adult stem cells. These are found in various tissues like bone marrow or fat, and these stem cells are more specialized than ESCs. They can differentiate into a more limited range of cell types specific to their tissue of origin. Let's say, for example, uh, bone marrow stem cells can become blood cells, while fat derived stem cells can become fat or bone cells. And while they are less versatile than ESCs, which are embryonic stem cells, ASCs offer a readily available source for potential therapies. So let's consider this analogy. We saw that they are more specialized. So what does this mean? So let's imagine them like workers who are great at building specific things. Some are very good at laying bricks. So you can relate this to bone cells. And some workers are very good at uh, some workers are very good electricians. So you can relate them to nerve cells, and others are great plumbers, right? So you can relate this to blood vessels. They are found in many tissues throughout the body that help repair and replace the worn out or damaged cells. So this is all about adult stem cells. Now let us jump to induced pluripotent stem cells. So imagine being able to take a regular cell from our body and turn it into a new type of stem cells. New cell type like heart cell or brain cell. So that's the amazing potential of iPSCs, which is induced pluripotent stem cells. Let's say, let's take the previous example only. We saw that some are very good at laying bricks, and some are good electricians, and they cannot become one other in ASCs. But with the help of IPSCs, they can be changed into one another. So let us see how. Scientists figured out a way to take a regular cell like skin cell and reprogram it using special factors. This reprogramming process essentially turns back the clock on the cell and making it behave like a stem cell again. And these reprogrammed cells are now called as iPSCs, which is induced pluripotent stem cells. Now let us discuss how to create iPSCs. We, there are four factors which are called as the Yamanaka factors, which are OCT4, SOX2, KLF4, and CMI. And they work together in symphony to reprogram the adult cells into induced pluripotent stem cells. Each factor plays a specific role in unlocking the cell's potential to rewind back to an earlier, much versatile state. So let's discuss each one of them. The first one is OCT4. This act as the master regulator, turning on the genes essential for pluripotency. And this also suppresses the genes that are associated with the cell's special functions. SOX, OCT4 is basically like the leader, and SOX2 is like assistant leader, which collaborates with OCT4 to activate the genes required for pluripotency. This also helps. with helps to opt for, and it also helps to maintain the self renewal capacity of the iPSCs. Now let's move on to the next factor, which is KLF4. This helps to erase all the specialized identity of the adult stem cells, and it promotes pluripotency. It works along the OCT4 to create an ideal environment for reprogramming. Now let's discuss about the last factor, which is c-MYC. This acts as a cell regulator, promoting cell division, which is essential for generating a sufficient number of iPSCs for our research. But it is important to note that um, CMI can also promote tumor formation, so its role needs to be tightly controlled. So let me ask you a question now. What happens if its role is not controlled? What is the name of that disease that uh, where the CMIC is not regulated properly? Yes, guys. So that disease is nothing but our famous cancer. In these cancer cells, this c-MYC is overexpressed and it leads to tumor formation as well. So this is the reason why it needs to be controlled or regulated. Now let's discuss the potential for organ creation using the stem cells. So once scientists have these stem cells, the magic begins. Different techniques are used to nudge these cells towards a specific fate, and this can involve using growth factors, which are signaling molecules that tell the stem cells to which direction they specialize in. And the next thing is called as scaffolds. So we'll be discussing about scaffolds in much detail in the next section. Scaffolds are nothing but 3D structures which are made of biocompatible materials that mimic the natural architecture of an organ and provide a framework for the stem cells to organize and grow upon. So, for example, collagen is also a biocompatible material and it can be used as a scaffold. So, this using stem cells helps us in minimizing organ rejection as well. Especially if we use iPSCs, there is no ethical considerations and it is the most preferred way. Yeah, sure. So let's yeah we there are two ways by which organs can be created in lab, which is 3D printing, which we'll be discussing in next section, and we'll discuss about scaffolds in next section as well, but I'll explain it to you now. Scaffolds are nothing but a supporting material. For example, if you cannot directly grow the stem cells on a petri dish and expect them to become the shape of an organ as such, right? So we create materials and we create scaffolds using biocompatible materials and we make these stem cells grow on them, and we give growth factors such that they replicate the function of our desired organ. So we get the function as well and we get the structure as well. Do you understand? Dhriti Jana. Okay. So scaffolds can be 3D printed or they can be made in another way also, which is you can take an organ, remove all the cells from it, so you will be left with the extracellular matrix, and then on that extracellular matrix you can grow your stem cells. I hope it's clear now. If yes, give me a thumbs up. Okay, guys. So let us discuss the protocols for creating iPSCs, which will be our last topic for today's session. The first process is cell isolation, and this process starts by isolating mature cells from a patient. This could be skin cell, blood cell, or any other type of cell, depending on their specific approach. So, what are general processes for isolating cells? There, there are many processes. Do you have any idea about it? What are the processes to isolate cells in general? Yes, there are many processes, and these are one of them. So, I'll be discussing only about two processes here, and I've given a link here. You can see more about the processes of cell separation from there. So, the first process which I'm going to discuss is fluorescence activated cell sorting. In this type of cell separation, the cells are tagged with fluorescent antibodies that bind to specific surface molecules on the cell. And then a laser beam identifies and sort the cells based on their fluorescence. The second type is the magnetic activated cell sorting. Here, the cells are labeled with magnetic particles coated with antibodies targeting specific markers in the cells. And then a magnet separates the cell population of interest. So, this is all about cell isolation, and you can learn more about it in the given link. So, let us move on to the second process, which is reprogramming, which we saw earlier. This is using the Yamanaka factors. But how do we introduce these Yamanaka factors into these cells? There are majorly two methods. The first one is using viral vectors. There are engineered viruses that deliver the genes encoding the Yamanaka factors into the cells. And this method is efficient, but it raises safety concerns due to the potential viral integration into the host cell genome. And the next process is non viral methods, and these are much safer than the viral ones. For examples, we can use plasmids, which are circular DNA molecules, and we can use messenger RNA, and we can use the protein delivery methods as well. The third process will be selection and expansion. After the exposure to Yamanaka factors, not all cells becomes iPSCs. Only a small percentage of cells will be successfully reprogrammed into iPSCs. These cells need to be identified and isolated from the cells which are not reprogrammed. So there are various techniques for this as well, such as observing the cell morphology and using specific markers which are associated with pluripotency. And using this, scientists can identify the potential iPSCs and they can create colonies with that. Once identified, these iPSCs are expanded in a culture to generate enough cells for further experiments or for therapeutic applications. The last process will be quality control because it's crucial to ensure the quality and safety of the generated iPSCs. Scientists perform various tests such as uh, the verification of pluripotency. This confirms that the iPSCs have the ability to differentiate into uh, different germ layers. So, can you name all the three germ layers in the chat? Yes, you're right, Gorija. Ecto, endo, and mesoderm. Yes, guys, all of you are right. So, the scientists verify that these iPSCs can differentiate into ectoderm, endoderm, or mesoderm. And they verify the potential of these iPSCs to become any cell type in the body. And they check for the genetic abnormalities as well because reprogramming can introduce mutations in the cells. So the cells need to be screened for any potential risk. And since the C mic can create tumors, we also need to regulate that factor so that the iPSCs which we generated doesn't have the risk of tumor formation. And since the organ is derived from the patient themselves, the immune system won't be recognizing them as foreign, which significantly reduces the risk of rejection. And this also reduces the need for immunosuppressive drugs, which can have serious side effects in us. So, we have come to the end of today's session. So, let's explore more about lab grown organs and 3D printing in next session, which is on Wednesday. So today's homework will be to download a software called as Fusion 360, which will be using to model the 3D model of trachea. So here's the reference link you can use to download that software. And let's have a sneak peek for session 2. We'll be discussing about the traditional methods for disease and drug testing, which is using animal models and using computational modeling as well. Then we'll be discussing about our hero, which is organoids. And then we'll discuss the success stories as well. And we'll be moving on to the 3D printing, where I'll be explaining you about the processes and how they help in for the creation of lab grown organs. And then we'll be having hands on experience on 3D modeling. Yeah. So that's it for today, guys. If you have any doubts, you can stay back. And I think some of you had some doubts with Arun's session as well. So those students can also stay back in the session. And otherwise, you can leave this session. I hope you found this session informative and exciting. So let's meet on Wednesday. Thank you, guys.
Hello. So this is just a reminder. So my the assignment two for my session, its deadline is today, so please submit it. And one more thing I noticed for that, some of you attached the wrong copy of the code. So you attached the codes for the questions that I did during the session. Assignment two has separate questions which you will find in the homework for certification channel. So if you have submitted the wrong questions by chance, please go through it again. Go through assignment two and submit those codes. And if you have any doubts related to assignment one or two, you can feel free to ask me now. Oh yeah, so for assignment 3, I've attached the code partially written, but if you want to write a code separately or in another language, you're free to do that.
Uh, Kushi, you can download it on mobile, but laptop will be much more convenient than using mobile. So you can look up to that. And the link which I shared is only for the laptop. If you want, I'll share the link for the mobile app as well.